predicting the risk of Sclerotinia in different crops. I'm actually doing a research project on predicting blossom blight and stem rot in seed alfalfa. I'm not working on canola, but because it's the same pest, it has all of the same models for disease development. Okay, so whenever we start talking about disease, I think a lot of you have probably heard Mike Harding often talk about the 3 pillars that a disease needs to get going. The first one is the host, the second one is the environment, and the third one is the pathogen. And when we start the season out, when we plant our canola, we already know a lot about the host. We know which cultivar we're planting, we know the density that we're planting the cultivar at, we know the history of the field that we're planting into, we knew when we had canola last. The 2 things that we can't control, that we don't know a lot about, is first the environment. It would be great if we could control the environment. And all that we can really do is monitor the weather and monitor the environment, and that'll help tell us what the risk of getting some of these diseases are. The third thing that we really don't know a lot about, and this is where disease forecasting comes in, is we don't know a lot about the pathogen. The best that we can do is guess how much pathogen inoculum might be present in the field based on crop history. And when we're talking about a disease like Sclerotinia or white mold, it has a really broad host range, and the sclerotia can survive in the soil for a really long time. I'm sure this is what every farmer researcher wants to see, is a giant bucket of inoculum in their field. So I'm carrying this over and making sure I don't spill it. But basically in here, we usually have summer students that spend the first couple months in May, early June, collecting buckets and buckets of this sclerotia, usually from bean debris. But if you want to come and take a look at this afterwards, this is what's left behind in your field, in the soil, after you've had a really bad white mold year. And it's this sclerotia that will survive in the soil for up to 5 years that will produce the initial inoculum for subsequent crops. And because sclerotia only produces one burst of inoculum, those sclerotia germinate, they produce something that's called an apothecia, which is kind of like a mushroom-shaped structure, except it's almost microscopic. And then you get the spores that are forcibly ejected from the apothecia or the mushroom structure that can travel quite a far distance and get into your canopy. So because we only get one burst of the spore release in a season, Sclerotinia is really an ideal disease to be able to use disease forecasting with. So what we want to do is try and measure the amount of spores that are being released into the air and then use that to predict your risk of getting white mold. So how can we measure something that is microscopic? I think there's been a few. Some of the older methods are doing petal tests. I don't know if everyone here has heard of that. So basically you go out, collect petals, plate them onto media, wait about 5 to 7 days, and you look at the results of your petal test, and you're like, yep, I had white mold, I should have sprayed a week ago when you did your petal test, right? The other way to do it is you can put plates out in the field, and again, a lot of researchers use that technique, but again, it takes 5 or 7 days for the fungus to grow out on the plates, so then again, you're like, yeah, I should have sprayed a week ago when my blossoms were first infected. So there's been a lot of research on using these spore samplers here, and we have 3 of them that we have out in the seed alfalfa growing region, and I just borrowed one for the day to be able to show you guys. And basically, how it works, there's a little air intake right here. There's a fan that is actively drawing in air, so this can sample large volumes of air over time. It gets sucked in, and then everything, all the spores, pollen, anything that's in the air gets deposited into these little vials. And this is called a multi-vial cyclone sampler. And basically you can leave it out running for a week, and you can collect samples in your vials every day. You can change it so that you collect samples every 2 hours. You come and change your vials out every 2 hours. For research purposes right now, we leave it out for a week, change the vials every week, and then look back and see, okay, what was the spore risk, last week or 2 weeks ago. And then the other thing is that once you get the spores into those vials, you have to be able to know what you have. The old method was looking under the microscope, which takes a lot of manpower. Who wants to spend every day counting spores under the microscope? The other thing is that the spores that Sclerotinia produce are nondescript. They're little round balls, really, when you look at them. And so there's nothing to distinguish that that is actually Sclerotinia. So the method that we use is advanced DNA techniques. So we can take the whatever is deposited into the spores, do a DNA extraction. And the old days, again, you'd have to then take it back to your lab, do a PCR. You know, it takes a fair amount of skill, pretty high-level expensive kits. There's something new that's just come out right here. So this is basically a lab or a field machine. Takes half an hour to test what's in your sample. It's actually running right now. I turned it on right when we got to the field with some samples. So if you want to come and look at the results, it's basically already telling me what was in those spores. So this is the idea that it's sort of a lab in a box idea, and these are getting cheaper and cheaper, and it's something that we're looking at to try and get rapid analysis of what's in your field. So what we do then with all that data that we're collecting, we're also collecting a lot of weather data, and so you want to be able to correlate when are the spores being released with different weather patterns, as well as the microclimate within your canopy. So it might be a hot, sunny day above the canopy, but inside the canopy at soil level, where the Sclerotinia, where the sclerotia are, it can be a lot more humid in there, especially if you've irrigated, which they do in seed alfalfa. So we're looking at the environment in the microclimate to be able to then correlate it to spore release, with the idea that if you can produce some really good models, eventually you can sort of do away with even having to collect spores and just look at weather variables to predict when you're going to get your spore release. And I should say that in Poland and in the UK, they actually have networks of these spore samplers out. I think in Poland they have them in about 10 different regions, and they actually produce a national network of risk for blackleg. They're working on developing something as well for white mold. And in the UK, I think they call it Brassica Alert, and they have a couple of different diseases there. But Syngenta basically sells a kit. You can buy a spore sampler yourself, and then you can test what your risks are. And they also do these national networks where they're telling you what your risk is. I think their bigger problem is ring spot, which we don't have here. So it would be really great if Canada or Alberta could sort of catch up with some of this technology that's going on, and if we could start to develop some of these national networks to predict the risk of disease, much like they have the Prairie Pest Monitoring Network, which tells you insects that are out there. It'd be great if we could move towards also looking at the diseases that are out there. And one of the great things about these spore samplers is that it's collecting everything that's in the air. So you might be worried about white mold in your Sclerotinia, but you're probably also growing wheat in another field. You could be growing potatoes if you're in the irrigated district. And so you can use the same sample from the same spore samplers to look at risk of late blight in potatoes, stripe rust in wheat. And so what we're doing, even though we're collecting samples for seed alfalfa, we're actually sharing our samples with some of the other pathologists, and they're going to test them for some of the other diseases that are there. So we can get a really good picture of what some of the spore levels are like in different crops as well. Okay, so I think that's all I had to say about the white mold monitoring. I didn't talk much about the disease. I can if you want me to get more into the disease cycle and symptoms of disease, but I was looking through Ken's canola crop and it actually looks really good. So it would be interesting to see if we had the spore sampler out whether we were actually catching any of those spores or not, because the crop here is really nice and clean, even though he tells me that he's been irrigating it to try and get disease to develop.
It's a good rotation. What's kind of the cost?
The spore sampler costs about $3,000. Right now, the only company that makes them is a company in the UK. It's called Burkhardt Manufacturing. There are some companies that are working on making these spore samplers in the States, but they're more for defense to be able to monitor bioaerosols, bioterrorism. So they're actually a lot more expensive. So Burkhardt is the company that has the patents and has really developed these spore samplers. This little machine is still a little bit more expensive. It's about $15,000, but the cost is coming down a lot. And you also, You don't need to have this machine to be able to run what they call an isothermal DNA detection. Basically, with a PCR machine, I don't want to get too much into the technology, but the temperature's ramping up and down, and that's what drives the reaction to be able to detect the DNA. With these new isothermal mechanisms, you basically just need a machine that will hold at 65 degrees. So there's a lot of researchers that are working in Africa that are just using basically a water bath to hold your samples at 65 degrees. The other thing that this machine does, though, is that it quantifies how much is there, so that's where the cost comes in. But there's other ways to be able to detect your DNA levels without having to use this machine.
You'd be able to, like, if you could have many spore traps, just one analyze it, right?
Yeah.
Well, how far apart would you have to put the spore trap collection pieces? Like if you had thousands, 5,000 acres of canola spread across 20 miles?
Yeah, generally I think you can put them about 50 kilometers apart and you get a pretty good indication of what the spore levels are like in that area. Again, there's a lot of research going on looking at how much spores actually spread, say, from within your field to neighboring fields, and so how accurate over long distances these are. But they've used these spore samplers in a program in Ottawa to try and monitor the soybean rust coming in, and so they have them spread over the border and they get a really good indication of what the spore levels are like.
So the ends of the meteorological stations of the province of Alberta would be a number of them would be really strategic as far as.
Yes, exactly. So if you could get spore samplers out too where all those weather stations are, you'd have a really good network of spore monitoring. But
Was the sampler that was about 15K or
the sampler is about 3,000. The analyzer is about 15,000. Yeah. And the other thing that they're doing a lot of research on in the UK, and this is actually what Syngenta sells in the UK for their kits, is something called flow cytometry. So almost similar to like a pregnancy test where you just have to dip a stick into your spore sampler or to the vial, and then it'll change color if spores are present. That doesn't quantify the amount of spores that are in the air, but it tells you whether they're present or absent.
Is Saskatchewan doing something similar? Were they doing something called Sclerotinia or Sclerotia depots or something like that?
Yeah, please answer because I don't know the. Yeah.
Yeah, they had buried some sclerotia depots around the province, and I don't know how well it's worked out this year. This is the first year of our project. I think that there were some issues because they were all measuring zero for quite some time, and they shouldn't have been. So I think there's some bugs that are getting worked out, but if it is something that should be done, I think it's harder to grow Sclerotinia in a depot in the soil than it is in normal field conditions. You wouldn't think it would be that hard, but very honest.
So they would go together, right? Because there's no sense sampling for spores if those flare bodies haven't put up the little mushroom and aren't releasing it anymore, right? So once you see those flare bodies starting to germinate into the last species, then you say, okay, now I have to start sampling for spores, right? So to me, they'd be quite complementary.
And if afterwards you want to come and take a look at the results on my little machine that you can't see from there, then please feel free.
Here's your parting gift.
Oh,
Thank you.
Thank you. I'll try not to spread it in your field.
Join me in thanking Syama. I think this technology actually shows a lot of promise, not only in monitoring disease, but what Ralph was talking about earlier is understanding the biology of the diseases as it relates to the type of climate that we have here as well. So I'm looking forward to seeing more of that work.